Hello, it's very nice to be here. I'm going to talk about the work I do at the European Bioinformatics Institute near Cambridge in the UK. And bioinformatics, as was briefly mentioned in the talk by Professor Gray yesterday, is the application of informatics and computational techniques to biological data, particularly molecular biological data. It's arguably the fastest growing field in biology at the moment. And before I talk about the future of the discipline, I'd like to begin by taking a journey back into the past, because the growing importance of bioinformatics is really simply a consequence, a logical consequence, of the major discoveries in biology over the last century and a half. And so perhaps the two first discoveries are discoveries related to inheritance, which is basically studies of why things resemble one another, and also studies related to evolution, studies of how things differ to one another. And the resolution of these two issues was, of course, provided by the groundbreaking work of Watson and Crick and others, with their discovery of this beautiful molecule DNA, this double-stranded molecule in which information is duplicated, which gives the ability for the molecule to self-repair and self-correct, and that in essence provides the stability property. But occasionally you get mutations, occasionally things go wrong, and you get changes in the DNA sequence. And it's this change, this mutation, this introduction of new variation, which explains why all of us are different to one another, and going back further in time, why we're different from apes and other forms of life. And this means that this is the basic tool of bioinformatics in some ways, the sequence alignment. DNA is, of course, a polymer. That means we can describe its structure of a given molecule as if it was a language. And the common evolutionary roots in this very consistent molecule mean that you can take the sequences of a given protein from many individuals or many species, and they're all essentially the same. But the mutationary process means there are key points of difference, and it's in that difference where where phenotypic variation derives from. And this means that from the very earliest days, bioinformatics was always going to be a computational science, because the value in these sequences and our capacity to infer from them depends absolutely on our ability to compare them. A sequence in its own is just a meaningless string of letters. When you get a series of sequences and line them up like this, you can identify where the variants are and correlate that with different function and performance and work out what's going on. So the very earliest days of what we might call bioinformatics were pre-computational. This is one of the first great reference manuals of the bioinformatics world. But things started to change when this thing came along. This is a device for sequencing DNA. When I was in the laboratory as a PhD student, this was what I used to use to sequence DNA. It resulted from the groundbreaking technological advances made by Fred Sanger, who found a very beautiful way of harnessing natural processes in order to actually tell us what the sequence of these molecules are. And as people began to produce DNA sequencing, so we began to get our first databases of DNA sequence, and they were computerized from the start, but in the beginning they were small enough that they could still have a printout and an annual report. Within a couple of years, the annual report was quite a bit larger, and sometime shortly after this, they stopped producing physical reports altogether. And I think one of the general points I want to make is that the reason that these databases grew so rapidly wasn't just due to the fact that the sequence was interesting or that the technology made it possible, but the fact that some of the people involved in setting up these databases at the very earliest stages saw it as very important to go and talk to the publishers and persuade the publishers that if you were publishing the results from a scientific experiment, you needed to actually publish your data. And meaningfully publishing your data meant putting sequence into a database. And in many ways, you can say that sequencing-related biology has really led the way in the culture of data sharing and data openness. And I think a lot of this culture was actually established by the very far-sighted people who founded these databases, and saw that there had to be very powerful incentives and drivers to get scientists to share their data, but when they did so, the true value of it would derive. So we've seen several exponential curves yesterday. This is one of them. One consequence of this explosive database growth was the founding of the EBI, where I work. And the EBI has in some ways a unique mission, which could be seen as blue and white collar science. Part of our job is to explore interesting, groundbreaking ideas based along bioinformatic analysis, but part of it is just plumbing. The necessary, but if unexciting, data management tasks which are necessary to turn open data into what in the latest buzzword in the field we call fair data, which has to be findable, accessible, interoperable, and reusable. And this really touches on some of what Sabina was saying in her first talk. The only start of it is to have a culture whereby people make their data available. To actually really make it reusable, you need to do a lot of work conceptually and infrastructurally in order to make sure that, okay, you can find the data, but it then takes you 5 months to actually work out what the format is, what it really means, and actually begin to be able to work with it. So that was, you could say, the first phase of DNA sequencing. The next phase came with this machine, which automated the process and allowed ambitions to rise. That led to the growth of genome sequencing. It's now been about 16 years since the human genome was deciphered. Further explosion of data, EBI gets a new building. And then what have we done since we've been able to sequence the human genome? Well, there's further technological advance. These are two critical machines in the history of that. The one on the left is an Illumina machine, which is an example of what we call next generation sequencing, which massively parallelized the generation of sequence and enormously reduced the cost of producing sequence. The picture on the right is a more recent machine. It comes from a company called Oxford Nanopore. You can see that it's very small. It's also very cheap, so relatively to get one set of sequence reads. It's also relatively quick. I mean, this slide, I think, is a bit of a corporate propaganda, but nonetheless, the basic fact is you can take your sample, you can do some very basic processing, you can put it onto the chip, you can have a sequence readout within just a couple of hours. So that's really quite an extraordinary thing, and this is perhaps a very interesting sort of exponential curve. You can see we've got a logarithmic scale here, and what that means is that when something is doubling in a fixed time period, it's represented by a straight line. And this is the straight line represented by Moore's law, by the growth in computing capacity. And in the earlier days of sequencing technology was generating the cost of generating new data was falling at around the same rate as the cost of compute capacity was falling. And with these next generation sequencing technologies, you've had an absolutely astonishing fall in the cost of producing a genome sequence, until we've now basically reached the long-aimed goal for being able to get a single genome, not for tens or hundreds of millions of pounds, but just for $1,000. So, it's great, we can sequence lots of genomes, but why have we developed these technologies and how are we using them? Well, one of the big projects of recent years has been the 1,000 Genomes Project data, which aimed to sequence not one individual, but 1,000. And in fact, which generated 25 times more data than was originally expected. And with this project, it was aimed to discover every common variant in the human population. And now we've moved on to even more ambitious projects. This is the 100,000 Genomes Project, and what's more, this project is based just in England. So the idea is that a large number of individually rare diseases, but which collectively affect still quite a high percentage of a population. In order to find genetic causes of rarer diseases, you need bigger sample sizes to get more power. And this project is both directly aimed at improving clinical outcomes, but also aiming to set standards more broadly for a new age in which medical care is much more digitalized than in the past. And of course, England is not unique in this. In the Faroe Islands, there's a project to try and sequence the entire population. So one thing we can do with these new technologies is sequence many more human individuals. Another thing we can do is sequence many more species. The bread wheat for has long been a very difficult species to sequence. It's actually a very interesting species because it results from the hybridization of ancestral species. So it's as if you had a species which contained the genome of human, chimp, and gorilla all in one individual. Well, there are some individuals who meet that description, but every wheat plant is like that. And this genome is extremely large and extremely repetitive, and for a long time has just been too costly to sequence. But in the past year, some relatively high-quality wheat genome sequences have become available. And in some senses, the technology has now reached the point where we can basically generate the sequence for anything we might want to. And so, as the holder of many of the reference databases which store this scientific information, EBI now has about 20 petabytes of non-redundant data and about 80 petabytes of data on disk in total. In our early days, we'd go and speak to the people at CERN and we'd say, we have problems with big data, and they'd tell us, no, you don't have problems with big data. But now the CERN people are actually more interested in us. And I think it's a very interesting thing in that different types of science have different modes for dissemination of data. So at CERN, you have a massive centralized facility generating enormous quantities of data which have to be spread out to scientists across the globe. In biology, a lot of these technologies are relatively cheap. In every laboratory now, you might have a sequencing machine, and the challenge is really to integrate this data and make sense of it. So one consequence of that is EBI has yet another building, but it's not in fact just at EBI that bioinformatics is growing, but right across Europe, and it's coming to infect every area of biology. So that's how biology has become a data science. What I'd like to do now is say a little bit about why we're sequencing so many genomes and how this will impact upon society, medicine, and agriculture. And finally, I'd like to wind up by saying a little bit about how we will try at the EBI, in our role of the maintainer of many of these key reference databases, to manage this data. So the point of my introduction was to try to give two messages: that sequencing is a key technology in all this, but really variation is the key component. And it's variation which explains how species differ from each other, how populations differ from each other, how individuals differ from each other, but also how diseased cells differ from healthy cells, how genes, cells, and organisms function. And this context of variation within the context of fundamental conservation is what's critical to that. Without variation, there's no evolution, no speciation, no genetic propensity to disease. So there are lots of areas in which these technologies might be able to impact upon society. For example, there are lots of diseases that are highly linked to genetic causes. If we can identify the putative alleles and sequence people at a young age to discover what their risk factors is, we can encourage them to change their lifestyles, and we can also target medicines towards people such that they're less likely to have negative side effects. Target discovery is a huge problem for the pharmaceutical industry. It takes 3 years to choose from millions of targets and then 12 years for a new drug to reach the market. And a large part of the reason so many drugs sell for so much over the cost of production is because of a huge investment that companies have to make in trying and failing through candidate drugs. But with extensive catalogues of reference information, we can help focus that process. And as I think Tony has already very ably explained, genomic sequence can also be hugely useful in the plant breeding process as well. So in natural populations, many variants are rare, especially those deleterious to survival. So a traditional approach to try to find the interesting variants involved trying to decipher causative relationships from patterns of inheritance in affected lineages. But phenotypes are often continuous and determined by numerous genetic loci as well as being rare. So medics would spend long time trying to identify families with propensities to disease, and the power of this tracking approach was very limited. But now we can use variants just sequenced across thousands and thousands of individuals, which make only minor contributions to disease, but identify those contributions through statistical correlation. And this is a sort of classic part of a technique. It's called a Manhattan plot. You scan the genome, you look for those sites of statistical correlation, and without any prior knowledge of what might be causing a given trait, you can identify the significant factors of association. And this is useful in medicine. It's also useful in food security. I don't think I need to talk about this slide because of Tony's introduction, but just say that these techniques are useful not just in breeding, but also in the development of new pesticides and monitoring and understanding the process of pathogen-mediated disease. And genomics can really improve the breeding process in many ways. We can perform the sort of phenotyping experiments of which Tony has talked and compare them with a panel of information about the genomic variation between different individuals. And thus, by bringing genomics and phenotyping together, we can identify those causative alleles. But also this enables us to track inheritance of those alleles through the breeding process. So even when we know what the relationship is, screening for phenotype is often complex and expensive, but once one's actually determined that a given genotypic marker is associated with a phenotype, one can screen genetically to find out that the key genes are present in the plant one wants to breed from much, much more easily than one can remeasure the phenotype. And this is even leading to people in plant and animal breeding building models where you can actually predict the total fitness of an individual based on its genetic background and really maximize the potential of the variation present in your breeding stocks to get a plant more acutely tuned to the environment. So, what do we do at the EBI about this? Well, what we do is we run a lot of databases. Sabina has given us an introduction to those databases. Although there are many organizations which run databases at the EBI, we run quite a lot of them. We run most of them in collaboration with many other important partners. Among the databases we run are among the least sexy but most important databases, the basic archives of molecular information from which many other resources which offer richer interpretation are built. And the resource I'm associated with is a resource called Ensemble. I'm not going to have much time to talk about Ensemble, but it was originally designed with the Human Genome Sequencing Project, and we now use it for all species from bacteria to fungi to plants. And here what we're really trying to do is to represent the information contained in DNA and about its impact upon biology. We integrate lots of data types. There's a lot of analysis to turn random bits of data, sequence you can pull out of the organism, and turn that into conceptual entities that are useful for biologists. Ideas like genes or regulatory sites, which can be inferred by doing analysis, analytics to use Gary King's word from yesterday, and turn this huge quantity of raw data into something that's more immediately accessible to scientists and useful for their work. So here's an example of just one of the very many numerous views we have within Ensemble. This is a plant example. What we have here is a phylogenetic tree showing the inferred ancestry of a series of genes from across about 22 different species of grasses. At the top in the colors you have the 3 genomes of bread wheat. So these are 3 genomes all from one organism which have come together again. And you can see this huge pattern of conservation. This is a stylized representation of the sequence alignment. And this is the sort of stuff we do as our bread and butter in Ensemble. We take reads, we turn them into gene models, we show the relationships between gene models, we identify the sites of variation. Another of our most important tools is actually something that's conceptually very boring. It's the variant effect predictor. If you've got some sequence from some individual that you've sequenced, you put it into Ensemble, the variant effect predictor compares that to the reference annotation. It tells you what the likely effects are of the differences in your sample on the well-known annotated reference genes and their function. And so this is really not rocket science, but in fact it's one of our most useful features. And I think it demonstrates something which is very, very important. With sequencing having become so cheap, we're moving from an age where people sequence for the purposes of discovery to find something new, to an age where people sequence for a purpose of assay, to find out where and when in the world something which is already known about is actually occurring. And this is very much linked to the spread of genomics from the laboratory into the field and into the hospital. But that assay sequencing means nothing unless you have reference data with which to interpret your results. And so our role is to ensure that the reference data is there and as clear as possible. So as we move forward, one big challenge is in improving our conceptual model of regulatory information. You can perform large-scale assays to identify which regions of the genome are controlling genes. You can perform large-scale assays to identify what the genes are, but actually linking them those two together isn't always clear. And as new techniques are being developed in order to make that linkage, we need to improve our interfaces to deliver that information to science. So, in my last few minutes, I'd like to talk about some of the challenges going forward. And one of the challenges is simply due to how do we store this data when this data is increasing in volume at a rate much faster than Moore's law. So this is a data accumulation slide. Again, we have a log scale on this axis. Again, that means a straight line represents exponential growth. Straight lines of this gradient represent doubling of growth at around the rates of Moore's law. And what we can see is that when you have a mature technology, more or less the rate of reference data is growing at around the rate of our computational capacity to store it. But when you have a technology explosion, the data volumes just increase much, much more rapidly than that. And that causes us problems, and that means we have to look at data compression. And this is some work of a colleague of mine, Guy Cochran. But I think it's an interesting slide to show that if you look, here is the growth of Moore's law, how much data you can store for constant cost. Here's what happens with biological data. It's growing much, much more quickly. So one solution is to compress, but compression only gives you a one-off boost. Suddenly you need 50% of the storage you did, but if in 5 years' time you were going to need 10 times as much more storage than you could afford, now in 5 years' time you'll only need 5 times more storage than you can afford. So compression would seem to be a fairly useless option in that it buys us a few months, but pretty soon we're beyond the limits of cost. But in fact, it's not all as despairing as this, because as we know more and more about the world, less and less sequence will be novel. More and more sequence will just be variants from what's already known, and that allows great ability for compression. We can also be very smart in terms of compression. These two images on the right are actually compressed to absolutely the same extent in terms of data capacity storage. But we can highlight our compression by storing most information about what's most interesting. And that means storing quality information, which can be important for interpreting data, around the sites of variation, and maybe throwing it away where a read merely confirms what was known already. And there's two factors, being selective in your compression and our growing knowledge of the world, are to date allowing us to keep track of this enormous growth of data and not yet bust our budgets. And these formats are now implemented in our archives and available to everyone. So my last few slides are about relevance to clinical practice and how we actually manage this transition from being very much in an academic research environment to having technologies which are put in use by doctors and in hospitals. And there are big, big cultural differences between the research environment and clinical practice. In research, we tend to use English as a language. Legal frameworks tend to be very lightweight. We have this big commitment to open data and scientific publication. We rely on grant funding. In practicing medicines, it tends to use national language. The legal framework is rightly and necessarily very heavy. You have very different systems between different countries. Privacy concerns, but also cultural concerns, means data is a lot more closed and a lot less widely published, and there's a contract system of funding. And to make it worse, in every country you have different systems of healthcare. So this is a slide just illustrating the difference between the British and the German system. If we were to add the American system or the Italian system in there, there's even more difference. So there's not even a consistent model across Europe and the world of what a healthcare system is. So are these problems going to be solved by the EBI and our other academic collaborators and the groups involved in building academic databases? No. Equally, is medicine going to be dependent on the reference data and definitive interpretations produced by research scientists? Yes. So to solve that, a bridge needs at least two anchors. And when we think about what long-term strategic goal is, we really feel that each nation is going to need to develop its own institutes for biomedical informatics, linking very closely with national healthcare systems. But these are going to have to interact with institutions like the EBI, which are capturing the cutting-edge research information that provides the reference data within which practical medical sequencing can be applied. So our institutions like us, our partners in this Elixir framework, which is a cross-European network of organizations involved in academic data, will handle research-style data and interactions with researchers, whereas the National Biomedical Institute will be responsible for exploiting this data and brokering data sharing in the medical context, where there are many more legal and privacy concerns. And of course, there will be grey areas. I mean, if you look at the 100,000 Genomes project in England, it's being very, very closely run, not by traditional academic institutions, but as part of the NHS. It's a research project, but it's really right on the cusp of this. And actually working out the correct legal, organizational, and practical ethical frameworks for dealing with this grey area is one of the major challenges we have over the next few years. And we're working to address these challenges together with a number of partners, both academic and commercial and healthcare providers, in an organization called the Global Alliance for Genomics and Health, in which we're trying to map out this new landscape and turn genomic medicine into a reality. So I'd just like to acknowledge the many organizations which fund us, and I'll be delighted to hear your questions later on the panel. Thank you.